schedule, Professor Heman graciously accepted our invitation, and we are truly privileged to host him here today. I'd also like to warmly thank Associate Professor Ahmet Acar from Middle East Technical University. Despite Professor Heman graciously accepted our invitation, and we are now truly privileged to host insights and expertise to this important topic we will discuss today. Now, to open our program, we are honored to begin with a keynote lecture by Professor Michael Heman titled Why Treatment Fails: Understanding Mechanisms of Resistance to Cancer Immunotherapy. Michael Heman studies the mechanisms that regulate the way blood cancers respond to treatment. His lab has developed tractable systems to acutely disrupt gene function in vivo and has used this approach to investigate the role of individual genes in modulating therapeutic outcome. The work has led to the identification of key determinants of cancer cells respond to frontline chemotherapy. Heman and his group have subsequently expanded this approach to assess the role of thousands of cancer-relevant genes and specific cancer mutations in drug response. Results from this work will better inform strategies to customize our current arsenal of anti-cancer agents to the specific constellation of genetic alterations present in a given tumor. Michael Heman earned a bachelor's degree in biology at Wesleyan University. He completed his PhD at Johns Hopkins University and went on to conduct postdoctoral work at Cold Spring Harbor Laboratory. He started his independent laboratory in 2006 at the Koch Institute at MIT, where he is currently a professor of biology. Dear Mike, we are very much looking forward to hearing about your groundbreaking work and perspectives on one of the most complex challenges in cancer therapy.
Thank you so much for that kind invitation. It's very nice to be here. We were fortunate to have Gulnahal join us at MIT for a research day in addition to her student Azin Leylik, where we were able to collaborate, I think, on a really important study that focuses on this issue of why cancers fail. I hope Gulnahal will talk about this work later because I think it's a really elegant study, and I think this is really an important idea, one that continues to plague us in the realm of cancer research. So my group has focused for a long time on this issue of why drugs fail. And to a great extent, we've really focused on conventional chemotherapies, the kinds of therapies that people are actually treated with mostly and have been for the past 70 years. And I think despite the fact that we have used these therapies for a really long period of time, we actually understand very little about why they work and why they don't work. And so we're not able to apply particular drugs to particular patients in ways that we would want to, and we don't understand when these therapies are actually going to work or not. And so the big question is, why do drugs fail? And so when I started, I think, and I think many people think, that resistance is caused by mutations. So cancers acquire alterations in their genome, and these cancers alterations fuel eventual resistance. And this is clearly the way it works in the context of targeted therapies, EGFR inhibitors and ALK inhibitors. Mutations are really important. But in the context of the therapies that we use mostly to treat cancers, this is likely not the case. And so this is work now done, I think, in a really groundbreaking study over a dozen years ago, looking at myeloid leukemia, so blood cancers. And what this group found was that if you treat a patient, the relapses to chemotherapy are clonal, meaning that only a subset of cells grow out after treatment. But the mutations that are present in these clonal relapses are not resistance-causing mutations. So it doesn't appear as if there are clear mutations that are present in tumor cells that promote the ability of tumors to survive therapy. And in fact, in work I won't describe, we've tried to recreate this process in our labs by using base editing alterations that introduce a lot of human alterations into cancers, and we find the same thing, that if you treat tumor cells that have mutations with therapy, you have clonal relapse, but we simply don't see mutations that we think correlate with resistance to the therapies that we use. So why are cancer therapies failing? So I thought very briefly I would summarize a really long set of experiments from our lab over the past 17 years that argue that perhaps it is more microenvironment, it's more context, than cell intrinsic change that mediates resistance. So I thought I would just present a few quick examples. And the first one was work that was done by a former graduate student in the lab, Luke Gilbert. And what Luke found was that if you treat tumor cells with chemotherapy, you induce DNA damage and you kill these tumor cells. But chemotherapy not only acts on tumor cells, it acts on cells next to these tumor cells, like the tumor microenvironment. So in Luke's case, he saw that these therapies act on endothelial cells, so vascular cells that are next to the tumor cells. And in response to chemotherapy, the endothelial cells actually secrete factors here like IL-6 and TIMP-1 that act on tumor cells that happen to be in that environment. And this promotes resistance to therapy. So these tumor cells and these tumor cells are exposed to the same amount of drug, but these tumor cells survive because they're in a space, a microenvironment that protects them from chemotherapy. Another example of how a microenvironment can be chemoprotective was work that was done by another graduate student, Simona Dahlin, who was looking at pancreas cancers. Now, pancreas cancers are really complex tumors. Only a subset of the cells in the pancreas tumor are tumor cells. Most of the cells are stromal cells, they are fibroblasts, they're immune cells. And she wanted to see, are these surrounding cells protecting tumor cells? And so she just looked at a single subset of cells in this microenvironment. She focused on stellate cells, which are a kind of fibroblasts that are activated in pancreas cancers. And she simply asked, do fibroblasts or stellate cells secrete things that protect tumor cells from chemotherapy? And so she did a simple experiment. She activated stellate cells in mice. She harvested these stellate cells, and then she let them secrete things in the presence of tumor cells and in the presence of the frontline therapy, gemcitabine, which is used to treat pancreas cancer. And what she found, in fact, is that stellate cells do secrete factors that protect cells from chemotherapy. So this conditioned media, or CM, contains something that promotes resistance to chemotherapy. And so she then did mass spec work to find out what that thing is. And curiously, the factor that stellate cells secrete is deoxycytidine. Why is that curious? Well, deoxycytidine is precisely the molecule that gemcitabine is a derivative of. So gemcitabine is a drug that's based on deoxycytidine. They're both taken in by the same transporter, acted on by the same intracellular kinase. And so you have a microenvironment in which stellate cells secrete a molecule that blocks the action of gemcitabine on target tumor cells. So it's a case of a microenvironment protecting a tumor. So the final quick example I'll mention is work that was done in collaboration with Scott Renalis looking at acute myeloid leukemia, work done by a couple of graduate students, Alex Miller and Yovel Lendner. And what they did is they essentially made a tumor that was resistant to a drug. And so how do you do this? You inject a tumor into a mouse, you treat it, you wait till the tumor comes back, you harvest the tumor, you inject again, you treat, and you have cycles of treatment until the tumor becomes resistant to therapy. And they saw something curious, and that is that if they actually treat this tumor over a long period of time, you get resistance to therapy in vivo, but you don't get resistance to therapy if you treat these cells in culture. So these cells are only resistant when they're in a mouse. They're not resistant if they're sitting on a cell culture dish. So it's not a cell intrinsic change that mediates resistance to the therapy. So why is it that they're resistant? Well, it turns out they're resistant because they actually go to a site of resistance. So we could use these circulation experiments to identify that these cells, once they're resistant, actually go to bone marrow faster than naive cells. And bone marrow is a site of chemoresistance. So they're resistant not because they have some mutation, they're resistant because they go to a site that is inherently more chemoresistant. And so these quick examples I mentioned because they're all examples where resistance is not mediated by a cell intrinsic change. I don't rule out the fact that cell intrinsic change can mediate resistance, but I think in the context of chemotherapy, and I'll argue today immunotherapy, this resistance more relates to microenvironment. So typically we think of resistance as an evolutionary process. We think about that with bacteria and viruses, that there's evolution towards resistance. But in the context of therapy, I think that resistance is really ecological. And so what I wanted to talk about today is very recent work, well, some a little bit older, but some very recent, that focuses on immunotherapy. And what I'll argue is that similarly, immunotherapy, some of these new drugs that we use to treat cancer, are also blocked by microenvironmental effects as opposed to just mutational effects. So as we think about how to make these therapies better, we need to think about treatment in relevant contexts. And so the class of tumors I'll talk about today predominantly are B-cell malignancies. So tumors of transformed B cells, blood cancers. So this is a leukemic bone marrow, a person that actually has lots of B-cell, cancerous B cells in their bone marrow. And if you treat this person with chemotherapy, these tumors are generally very responsive. So this is really a more normal looking bone marrow that doesn't have really any recognizable tumor cells. But in many cases, patients relapse, and they relapse with tumors that are harder to treat. So these are treated with cytotoxic chemotherapy and targeted therapy. But once these tumors come back, we now treat them with immunotherapy. We treat them with antibody therapies. We also treat them with CAR-T therapies. And so I'm going to talk about both of these therapies and mechanisms of resistance to antibody therapy and CAR-T therapy. So starting with antibody therapy, this is work that was done by a former postdoc in the lab, Christian Palash, who wanted to develop what he called a humanized mouse model to study cancer therapy. So what is a humanized mouse model? A humanized mouse model is a model that is essentially a human tumor in a mouse, derived from essentially normal human cells. And so how do you generate these? You actually reconstitute a mouse with human hematopoietic stem cells, and then you modify them with cancer-altering mutations. The benefit of using this kind of model is that we can model therapies that use human-specific antibodies, and we can combine chemotherapy and immunotherapy in ways that, that is done in human cells. And so in this case, how do you do this? Well, what Christian did is he expressed two oncogenes, in this case MYC and BCL2, which are common oncogenes in human B-cell cancers, along with a fluorescent protein GFP, under the control of a CD19 enhancer. So this is a B-cell enhancer that will direct expression of these target proteins into the B-cell compartment, use viruses to infect human hematopoietic stem cells, and then transplanted these stem cells into an immunodeficient recipient mice. And so this essentially recreates a human-like hematopoietic system in a mouse. And so what happens if you generate or if you reconstitute mice with these modified stem cells? Well, the tumor that Christian was trying to create was a tumor called a double-hit lymphoma. Double hit in this case just means it has two oncogenes. It has MYC and BCL2. This is a particularly difficult kind of tumor to treat in humans. It's really refractory to most chemotherapy, so there's overall poor survival in the absence of immunotherapy. And in fact, if he put MYC and BCL2 in, he actually got very similar tumors. So the red line here are mice that die within about 100 days after getting GFP, MYC, and BCL2, GMB. They don't get tumors if you just have MYC or BCL2 or GFP. And these tumors resemble, to the extent that a mouse tumor can resemble a human tumor, they resemble the human disease, and they're really resistant to the kind of therapies that we use. And they also phenotypically resemble the disease. You have big tumor cells in the bone marrow, you have big spleens, you have infiltration into the brain, which occurs in many lymphoma and leukemia patients, and you have chromosome abnormalities that are characteristic of the human disease. So again, the reason why he generated this is that he could use human-specific therapies and test resistance to these therapies in these models. And so the therapy that he decided to use was this therapy called alemtuzumab, which targets a cell surface protein called CD52. And so what happens if you treat these mice with alemtuzumab? Well, it has an effect, but it has a reasonably limited effect. So this red line are tumor-bearing mice that are treated with this monotherapy, and yes, there is a difference, but these mice all relapse. The disease comes back in all of these mice, so it doesn't work very well. It works a little bit. So how does alemtuzumab work? Well, it works by promoting macrophage clearance of target tumor cells. So how do we know that? Well, we know that because if we eliminate macrophages, we don't get an effect. So this is looking at the killing of target tumor cells on a log scale. And what you can see is if you add alemtuzumab, you get lots of killing of tumor cells, but you don't get killing if you eliminate macrophages. And you require both the FC portion and the FAB portion of this antibody. If you just have the FAB portion, you don't get killing. So how does it work? Well, you have an antibody, alemtuzumab, that binds a target tumor cell, and then you have macrophages that recognize the FC portion of this antibody, and they clear the target tumor cells. But this isn't working very well because you have only a short survival. So why do you get failure? So to try to investigate this, Christian looked at response in a variety of organ systems. So for example, if you just look at the blood, you have lots of clearance of tumor cells. So the therapy is working in blood. It's also working in the spleen and in the liver. These are sites where blood cancers go and infiltrate. But it's not working in the bone marrow, and it's not working in brain. So the vast majority of tumor cells persist in the bone marrow. This is actually a site of chemoresistance. So here we have another example of how a microenvironment, the bone marrow, is protecting tumor cells from therapy, in this case, immunotherapy. I should also say that it's not working in brain, and I think it's not working in brain for another reason. But the vast majority of cells persist in bone marrow. And so you next wanted to see, is that because the therapy isn't actually making it to the bone marrow? So here he used a labeled antibody, this Alexaflor antibody. The tumor's GFP positive. The antibody is Alexaflor 750 positive. And so he finds that, in fact, it gets to spleen. It doesn't get to brain, but it does get to bone marrow. So the therapy's getting there, it's just not working. So we know that we need macrophages to kill these target tumor cells, but they're unable to clear these tumor cells in bone marrow. So how do we actually overcome this resistance? And so one of the advantages of having this kind of model is that we can combine different therapies together to see if we can get an improved effect. And so, for example, he combined alemtuzumab with GM-CSF. This is a molecule that will boost macrophage production. And it doesn't do very much in this context. He also added radiation. Radiation doesn't do a great deal. There's not much of a combinatorial effect. But curiously, he saw real synergy if he actually added the alkylating agent cyclophosphamide. So again, this is a log scale, and we have almost 5 logs of enhanced killing if we add this agent cyclophosphamide, this alkylating agent. And in fact, we cure mice when we do this. So this is just a sort of a snapshot, but these mice that survive here survive considerably longer. If we just use the single agents, we get only a moderate effect. The combination actually has a curative effect. And these mice that are dying here aren't dying because of their tumor burden, they're dying because of toxicity. And these are both toxic agents. Cyclophosphamide certainly is, but this antibody is also a highly toxic agent. But Christian found that he could actually reduce the doses. So he can go from here 300 milligrams per kilogram down to about 100, which is a non-toxic dose, and still get this synergistic effect. Now to try to figure out why these agents are working together, he looked at essentially the dose regimen and found that you actually have to add these agents pretty close to one another. So you can only give them about 1 day apart and get the synergy. If you separate that, you don't get the synergy anymore. So why cyclophosphamide? What is cyclophosphamide doing? Cyclophosphamide is a very old chemotherapy. In fact, it's one of the first that was developed. It was developed in part because of work by Goodman and Gilman, who were working on chemical weapons that were used in World War I and in World War II. And these nitrogen mustards were then derivatized to generate cyclophosphamide. But it was known very early on that cyclophosphamide has unusual effects. It has vesicant effects. It has immunostimulatory effects. In people exposed, you get this kind of blistering. So you wondered, well, maybe it's actually doing something that's unexpected, that is not just on DNA. And so he did, I think, a clever experiment that was similar to the conditioned media experiment I described before. Here he treated mice with cyclophosphamide, he harvested cells, and he just let them secrete things into the media. And then he added this conditioned media to tumor cells plus macrophages plus alemtuzumab. And just with a little dose, he actually saw an increase in antibody killing. And this can hopefully be seen in this movie here. So this is a movie with green tumor cells and unstained macrophages next to them. And if you look in the middle, you'll see when we add this conditioned media that we actually improve the ability of these macrophages to engulf target tumor cells. This one in the middle will eat, I think, 2 more that come from the bottom. They eat 4 and then they die for some reason. They get full. But this conditioned media from tumor cells, these tumor cells are secreting something that makes macrophages better able to kill target tumor cells. So what are they secreting? Well, they're secreting classic macrophage activators like CCL4, IL-8, VEGF, TNF-alpha. So when you actually have these factors, macrophages not only get activated, but they get recruited into bone marrow, this site of chemoresistance. We see this here. In the absence of cyclophosphamide, we have green tumor cells. When we add cyclophosphamide, we get red macrophages that traffic into bone marrow. So we have essentially yellow combination of green and red of macrophages consuming these target tumor cells. And we see this in patients as well. If you treat with cyclophosphamide, you get this influx of macrophages into the bone marrow. You can see this here. If we just treat with PBS or another chemotherapy, doxorubicin, we don't have macrophages in bone marrow. If you treat with cyclophosphamide, you see this influx of F480 positive macrophages into bone marrow. And so not only are you getting more macrophages into bone marrow, but the macrophages are also different. They're activated macrophages. And just an example of this is work we did with David Weinstock, where if you actually look at macrophage populations, you see a new population, this number 4 population by this T-SNE plot, that is characteristic of cyclophosphamide addition. And just to mention one example of how they're activated, they start down-regulating decoy receptors, so receptors that block FC interaction with macrophages, and they upregulate FC receptors that specifically interact with macrophages. So just a summary of what we think is going on is that the bone marrow, in this case, is a repressive microenvironment. It's blocking the action of this kind of immunotherapy. So here we have a leukemia cell, and we have macrophages that are expressing inhibitory FC receptors. We also have other factors in the bone marrow that block activation. But if we treat with cyclophosphamide, we get this secretory effect that releases these factors like VEGF and IL-8 that activate macrophages, promote their recruitment into this inhibitory microenvironment, and then allow them to target these tumor cells. So importantly, this is a case where a microenvironment is mediating resistance to an immunotherapy. But it also says that there are strategies to overcome this kind of resistance. In this case, I think an interesting strategy because it uses a very old chemotherapy, one that is used normally in the context of treating B-cell cancers. But here, in combination with immunotherapy, seems to increase efficacy. This occurs not only in mouse humanized models, but it also occurs in PDX models. So if we treat human-derived, so human patient PDXs, we see a similar combinatorial effect here in 2 cases where this combination therapy shows a real synergy over single-agent therapy. And we're able to work with a few investigators over at Dana-Farber Cancer Institute to try this in a clinical trial. And here you have a patient in the purple line that is progressing just on alemtuzumab alone, but if you give this patient cyclophosphamide, in this case, you seem to drastically increase the efficacy of this therapy. So a subset of patients are highly responsive to this combination. This is also, though, a reasonably toxic combination therapy. And in B-cell malignancies, most patients are now treated, if they fail on chemotherapy, with alternative immunotherapies like CAR T therapy. So the second example of what I wanted to mention is work that we perform looking at basic mechanisms of resistance to CAR T therapy. So in a lot of patients that show tumor relapses in B-cell malignancies, both in lymphomas as well as multiple myelomas, these patients are now treated with CAR T therapy, which I'm sure all of you are familiar with. But just to provide a brief summary of how CAR T therapy is performed, you start by harvesting T cells from a patient and then modifying these T cells by retroviral infection or lentiviral infection via the introduction of a CAR, so a chimeric antigen receptor. These CARs are specific to a particular target on the malignancy of interest. In B-cell malignancies, we generally target CD19. These modified CAR T cells are then reintroduced into the patient. So this is an emerging therapy. In the United States, it's a very expensive therapy, so it's $1 million to $2 million per patient to treat patients. So I think it's important to think how we can improve these therapies, how we can make them cheaper, how we can make them more accessible to a lot of people. So from a blown-up perspective, this is what CAR T therapy looks like. We have a T cell in which we've introduced a chimeric antigen receptor. This CAR targets a particular antigen on a tumor cell. In the case of B-cell malignancies, we generally target CD19, as I said. This tends to be very well tolerated in patients. We don't know exactly why B-cell malignancies are more responsive to CAR T therapy than other malignancies. One of the benefits of treating a B-cell malignancy is that people can live without B-cells. So if you completely eradicate some of these normal B-cell compartments, they can survive and they can be infused with antibody. You can imagine if you have somebody with a brain cancer, you can't treat them with a brain toxic CAR T cell, so it's a lot more challenging to find the right target. But certainly it works well in the context of B-cell malignancies where there are very high initial responses, so amazing responses in patients that have failed on chemotherapy, but long-term many of these patients relapse. So the therapies can be reasonably short durations. And so the question is, why do these therapies fail and how can we make them better? And so one of the strategies that we frequently use in the lab is to actually perform genetic screens in vivo for modulators of therapeutic response. And so this is a screen that was performed in part by 3 graduate students in the lab who performed an in vivo whole genome screen for modulators of therapeutic response. And so the model that we used to perform this whole genome in vivo screen was this BCR-ABL positive model of B-cell acute lymphoblastic leukemia, or B-ALL. This is the most common cancer that is treated with CAR T therapy. So this is a mouse version of that cancer, expresses BCR-ABL, so this fusion oncogene. But this has real advantages as a screening model. And I just wanted to talk briefly about why we choose this kind of model to perform in vivo genetic screens. The short answer is that this is a highly transplantable model. And by highly transplantable, it means that we can transplant very few cells and we can get a tumor in a recipient mouse. We can essentially inject as few as 1 or 2 cells and get a tumor in a recipient. And this is really important because what we want to do is we want to introduce very complex libraries of alterations into these tumors. So we want to introduce 10 or 20,000 different alterations where each cell only has 1 alteration and then transplant these as a bulk into recipient animals. So we need lots of different cells to engraft in a recipient if we want to screen lots of different perturbations in the context of individual mice. So we need a highly engraftable system. So not only is this really highly engraftable, but it's treatable by the kinds of therapies that we want to model. So it's treatable here by CAR T therapy. Here we've introduced a couple doses of CAR T cells, and here we get a moderate response. And so for screening, this is sort of where we want to be. We want to know that therapy can get better or it can get worse, depending upon the alteration that we put in. So we want to introduce very large libraries and essentially assess what is the impact of particular genetic perturbations on therapeutic response. And so the library that we constructed with the Broad Institute, which is across the street, is a pool-based library. What does a pool-based library mean? It means that we divided guide RNAs for CRISPR-Cas9 targeting into about 48 different subpools. So each pool has about 2,000 different guide RNAs. And we introduced essentially collections of pools into recipient mice. So we can't screen the entire genome at once, but we can screen multiple pools in individual mice. It would be very expensive if you wanted to screen one perturbation per mouse. We want to screen lots of perturbations, but we need to figure out just how many perturbations we can screen in individual mice. And so the way that we figure out just how many things we can screen, the simple way is we do this kind of GFP dilution experiment. So we have increasingly dilute percentages of green cells that we inject into mice. So we inject 5% or 1% or 0.1% or 0.01%, and we see how low can we go and still represent a certain percentage of green cells. And so we know we can represent about 0.01% GFP cells. So what does that tell us? It tells us we can screen about 10,000 things in an individual mouse. So that's our limit. We can screen pools of guide RNAs. And so this is the screen design. It's a complicated slide, and so I don't expect you to sort of follow it. But the bottom line is that we modify tumor cells and we inject them into mice that express these guide RNAs. And then we add CAR T cells, and we look to see what promotes resistance to these CAR T cells and what promotes sensitivity. And as I mentioned, we can screen a lot of things. So this just says that we can actually introduce thousands of guide RNAs and track them in individual mice. And just as a little bit of a sanity check, we looked at essential genes, so genes that were characterized as essential via the DEP map, and we know that if we put in essential genes, these essential genes drop out in vitro and in vivo. Sorry, that should say in vivo. So we know that the screen is sort of working the way that we think it does. So what does this kind of screen tell us? Well, at the end of the screen, we actually get a list of guide RNAs that enrich and a list of guide RNAs deplete. And if they enrich, it means that they promote resistance to CAR T therapy. So this is CD19 targeting CAR T therapy. And so the guide RNA that targets more than anything promotes more resistance more than anything is CD19. So this is expected. If you knock down the target of the CAR T cell, the CAR T therapy no longer works. So by far, the most enriched guide RNAs are guide RNAs that target the target of the CAR T cells. We see a lot of guide RNAs that deplete, and these are ones that we're particularly interested in because these represent putative drug targets, things that when you inhibit them may sensitize a cell to CAR T therapy. And so there are numerous categories that include regulators of the synapse between a CAR T cell and a tumor cell, drivers of B cell identity. This, I think, is an interesting class. If a cell is no longer a B cell, then it will no longer express CD19. But the category I wanted to start mentioning is this interferon gamma JAK-STAT signaling pathway. And I mention it because it highlights, I think, a really important thing about both the biology, but the difference in performing a screen in the right microenvironment and a screen in the wrong microenvironment. So if we look at things that deplete, guide RNAs that go down in vivo versus guide RNAs that go down in vitro, we actually see very little overlap. If we do a screen in vitro versus a screen in vivo, we see very few things that actually deplete in both contexts. And I think, again, it highlights the importance of screening in the right context. I think you'll hear in the next talk about organoids, and that biology, I think, is also incredibly different. If you screen in organoids versus just 2D culture, you see very different biology. So I think it's really important to look at organoid models and additional models in this kind of screening approaches. But I'll highlight particularly the JAK-STAT signaling pathway because not only do we see a difference in the JAK-STAT signaling pathway in vitro and in vivo, we actually see completely the opposite effect. So that's really blown up here. So if we do a screen in vivo, we see the JAK-STAT interferon gamma pathway. If we lose it, highly sensitizing to CAR T therapy. If we do this screen in vitro, we see completely the opposite effect. We see loss of this pathway promotes resistance to CAR T therapy. And so what is the JAK-STAT signaling pathway? Well, it's sort of schematized here. I think probably a lot of you are familiar with. You have cytokines, and cytokines interact with a receptor. So for example, interferon gamma will interact with the interferon gamma receptor, and it will signal through JAK-STAT signaling downstream to transcription factors that mediate a lot of cellular responses. And it's not surprising that we see the JAK-STAT signaling pathway as important for an immunotherapy because it's really well established in a lot of important studies that JAK-STAT signaling is critical for responses to checkpoint therapy, so PD, PD-L1, CTLA-4 therapies. It's known that if you knock out JAK-STAT signaling, you mediate resistance to these therapies because, among other things, these are important for the expression of MHC proteins on cell surface. So it's important for normal T cell killing, but we actually see the opposite effect. So we see something very different. We don't see promoting resistance, or we see it only in vitro. We actually see it really sensitizing to therapy. And so just to confirm this result, we do this kind of validation approach. And what I mean by validation is we look at single alterations and we see, do they have the same effect as they had when we did the screen to begin with? And the way we do this is we actually mix modified cells. So say we knock out the interferon gamma receptor, we'll have interferon gamma receptor knockout cells, and we'll mix them with cells that are normal, and we'll actually let them sort of fight it out and see who wins and who loses. And if we do that, our results are really consistent with our screen data. So if we knock down interferon gamma or JAK2 or STAT1, these cells really deplete relative to normal cells if we give CAR T therapy. So again, it's consistent with the fact that these tumor cells are really sensitizing to therapy, and if we put them in as pure populations, we can actually have almost curative outcomes with STAT1 and interferon gamma knockout cells, and an effective sensitization to therapy if we knock down JAK2. So loss of this pathway is really sensitizing to therapy in vivo, but if we do the experiment in vitro, we actually see the opposite effect. We see these cells enrich. And so just really briefly, why do we see this really big difference if we do an experiment in a mouse or we do an experiment in cell culture? Well, it actually took us a long time to figure out what was happening. And this is certainly something that reviewers want to know because the consensus in the field would say this is an important pathway to get a response to therapy. And so it turns out that if we do experiments in cell culture, we get very, very, very high levels of interferon gamma, so non-physiological levels of interferon gamma. And so the loss of this interferon signaling pathway is necessary to promote resistance to this super high level of interferon gamma that you don't see in mice. In mice, we actually see that resistant tumors at the end of therapy actually tend to have reasonably high levels of interferon gamma. So tumors that we would consider to be hot tumors, immunologically hot tumors, are actually resistant to CAR T therapy, and we see that in people as well. So what's going on here? So to get some clue, we actually looked at another hit that was derived in the screen, this H2T23, also called HLA-E. This is also a real sensitizer to CAR T therapy. And what it does is it sits on the surface of tumor cells and it actually blocks the action of both NK cells, and we think it blocks the action of CAR T cells. And in fact, JAK-STAT signaling is essential for expression of this surface protein in response to interferon gamma. So if you knock out STAT or JAK or interferon gamma, you don't get HLA-E expression. So this represents a therapeutic target. And in fact, we can put in an antibody that targets HLA-E, and we can really sensitize tumor cells to therapy. So we have an in vivo specific resistance mechanism, And if we knock out or we suppress HLA-E, we can really sensitize to CAR T therapy. So what's going on here? So we have this simple interaction between a CAR T cell and a target tumor cell. The CAR T cell expresses a CAR, the tumor cell expresses CD19. And typically when this happens, the CAR T cell will just kill the target tumor cell. It will secrete perforin and granzyme and will eradicate this tumor cell. But we also know that when these CAR T cells get activated, they secrete interferon gamma. This interferon gamma interacts with the receptor on the target tumor cells. It signals through JAK-STAT, it activates HLA-E, and HLA-E gets expressed on the surface of the cell, and it in fact blocks the action of the CAR T cell through this inhibitory receptor. So I've been working in the resistance space for a long time, and this is a weird kind of resistance. So it's not you have some mutant cell, and this mutant cell resists therapy and it grows out. It's not evolution, it's not selection, it's adaptation. The therapy itself, the CAR T cell, is promoting its own resistance. It is releasing something that causes the tumor cell to change and present an inhibitory receptor that blocks the action of the tumor cell. So this is something that occurs in the tumor microenvironment. It is an adaptive change that allows the tumor cells to persist in the presence of this therapy. So it's another example of a microenvironment mediating resistance. So this is a case where the microenvironment is blocking these interactions in the relevant microenvironment and blocking the action of the tumor cell. And this is an adaptive response, a response that is mediated by a change in the tumor cell in the presence of therapy, and can be overcome if we actually block this microenvironment effect. So I'll finish by just presenting, I think, one other interesting set of biology that I think we learned from doing these in vivo experiments that highlight another really unexpected mechanism of resistance. And so one of the things we noticed that was curious is that if we put tumor cells together with CAR T cells, very shortly after putting them together, we see the loss of antigen from the target tumor cell. So the target tumor cell expresses CD19 initially, but very soon it stops expressing CD19. So here we see loss of CD19 from target tumor cells. But if we take the tumor cells out of the mouse and we put them in cell culture for a period of time, in the absence of CAR T cells, they start expressing CD19 again. So it's not that they've mutated it, it's not that they've eliminated the protein, they still have it, they're just not expressing it on the surface. So in the presence of CAR T cells, you have red tumor cells but no blue CD19. If you put them in cell culture for a period of time, they start re-expressing CD19 on their cell surface. So what's going on? Why is it that they're losing the CD19 from the cell surface? Well, what's happening is I think unexpected biology that's called trogocytosis. And what trogocytosis means is that the CAR T cells are literally ripping CD19 off of the surface of the target tumor cells. So here we have images where we have green CAR T cells, we have red leukemia cells, and if you look at CD19, the CD19 is not on the surface of the tumor cell, it's actually on the surface of the CAR T cell. So the CAR T cell has ripped the antigen off the target cell and it has re-expressed it on its own surface. And curiously now, CAR T cells start recognizing one another because the CAR T cells will express the CAR and they'll also identify CAR T cells that are expressing the CD19 antigen. And you can imagine, and I'll mention in a couple slides, that this creates a problem. This is an effect that we actually see in vivo, so if we do these in vivo treatment experiments, we see lots of CD19 here on the surface, not of tumor cells, but on CAR cells themselves. And so what happens when you actually have this kind of interaction is the CAR T cells start killing one another. So if you look at different ratios of CAR T cells to tumor cells, we start seeing loss of CAR T cell survival. So we would call this fratricide. So they're killing each other, or brother is killing brother. I don't know why it's brothers and not sisters, but they're fratricide that is leading to the absence of CAR T cells. And interestingly, a couple hits that we got in our screen, these proteins FIDM2 and PAC2, seem to be directly important in this trogocytosis. So if you lose these proteins, you don't get trogocytosis, and you see essentially more CAR T cells in the microenvironment. So we think that one of the ways that CAR T-cell therapy is actually failing is that you actually have the therapy itself, the tumor cells, that are mediating the death of the CAR T-cells that are trying to target them. So we see this trogocytosis occurring at the tumor cell CAR T-cell interface, but cell killing, the therapy itself, is killing itself. It's limiting its own efficacy. So we have induction of an adaptive inhibitory response. We also have acquisition of antigen, which is mediating death of these tumor cells. So again, these are both non-genetic mechanisms of resistance. And I want to be clear again, I think that there are genetic mechanisms of resistance. But as we think forward to these new therapies that we're developing, I think we run into the same problem as with the old therapies that we've been using, which is that we don't particularly understand basic mechanisms of resistance. We attempt to apply to all patients. And so agents like PD-1 or PD-L1 inhibitors that have turned Merck into the biggest drug company in the world, Merck in general has no interest in identifying who's going to respond and not respond to its therapy. It doesn't stratify cohorts, and it doesn't look at mechanisms of resistance. And so I think this is really important in academic communities that we think about why these therapies are failing, what are basic mechanisms, and how we can actually overcome them. And then we would argue again that I think initial responses to a lot of these therapies rely on this kind of constriction point where the microenvironment is protecting, and then you have outgrowth of cells that may not have the mutations that we expect them to have. I want to thank you for listening to me. These are the people in the lab that have done all of the work. We have a number of collaborator labs in our area. These are the really talented people in my lab. These are the people in my lab. They're in front of this really amazing picture that was developed by a really talented graphic designer, Aslı Gültener, who's here in Ankara, actually, who does a lot of art for our lab. So thank you again, and thank you for the invitation.
I don't know if my voice is counted. So I would like to thank again to Mike for his inspiring and insightful talk. We would love to have your questions.
Thank you so much, Mike, for the enlightening talk. So this, out of curiosity, just to get your thoughts, this non-genetic resistant mechanism, do you think they are the one leading to persistent phenotypes? And my second question is, do you think we can make such a generalization that these non-genetic resistant mechanisms are more reversible than genetic-driven resistant mechanism?
Yeah, I think both very important questions. So I think I should probably clarify that one of the things that in this case, when I'm talking about genetic mechanisms, I'm thinking most of emergent mutations that occur. You have a heterogeneous tumor. You have a subset of cells that have some alterations. They grow in the presence, out in the presence of therapy. And again, that's the way we think about targeted therapy, and that's clearly what works. You have a BCR-ABL mutation. That's what causes resistance to a BCR-ABL inhibitor. You have an ALK mutation. You have an EGFR inhibitor. You have other mutations, bypass mutations for BRAF inhibitors. This is absolutely what happens. The intrinsic mutation state is important. So the mutations that actually got a tumor there to begin with are going to determine how much drug you have to use, whether it responds or not to a variety of therapies. So those cell intrinsic states, I think, are incredibly important in that sense. So the persister biology that emerges, I think, is in the context of chemotherapy. I think it is a combination of essentially the inherent sensitivity or response of that tumor to therapy plus microenvironment. And I think microenvironment is going to play a key role there. And in some senses, it's common sense. For chemotherapy, we're always driving therapy to the maximum tolerated dose. And if you have a microenvironment that causes a 2 to 3-fold increase in resistance, that inherently is going to change the treatment landscape in that microenvironment. And so you're going to have persistence in that microenvironment, the way that we would operationally define persistence. In terms of the creation of a persister cell, the way we think about a persister cell, that may be different, right? And I think that certainly in the context of targeted therapy, I think that's probably mediated by resistance, mutations, or bypass alterations. But I can also imagine that microenvironment is going to also activate tolerance pathways that are going to allow for the persistence of those cells. So I would guess for immunotherapy and chemotherapy, a lot is going to be driven by the microenvironment. Maybe a different landscape for targeted therapy.
Wonderful talk. Is it possible to predict this resistance before therapy?
I think that one of the things that we should think more about is how tumor states drive tumor tropism. So how inherent states of the tumor promote the ability to survive in particular microenvironments and receive signals from those microenvironments. And that may be what we can predict. That there may be an inherent tumor state that makes it more likely to receive receptive cues in bone marrow or more likely to go to the brain. And this, I think, is, in the context of tumor metastasis, solid tumor metastasis, is really a pressing issue because it may not be mutation that drives metastasis. It may be inherent states that allow a tumor cell to survive in that microenvironment. And once it gets to that microenvironment, it is much more difficult to treat with systemic therapy because of those factors. So I absolutely think that that's something that we need to look at, how receptive cells are to those microenvironmental survival signals.
Thank you very much for your excellent presentation. My question is, you have given several different tumors that can behave differently and that have different oncogenic mechanisms and different of transformation capacity, such as BALL or multiple myeloma or malignant melanoma. They are very aggressive tumors that for BALL, if you treat in a correct way or if you have a chance, then you can cure the patient. But the thing is, you have to. But some patients you cannot be able to cure, and it relapses. But these relapses and cure issues, we track the patient for measurable residual disease in order to treat or to add some other things and to give some growth factors, for example, to increase the immune microenvironment. These are all the things that you behave for the tumor to detect the tumor and then decide when you give the drug. But CAR T cells that you mentioned, that you spent a lot of money, but the success is not that much high every time. My question is it for certain, for example, BALL, or I don't think that it could be possible maybe for multiple myeloma and or malignant melanoma, these are more aggressive. For BALL, at the first, how you can screen the potential of the tumor, whether when add some more things for getting more success to the treatment. So could you predict beforehand, searching the microenvironment, for example, or the tumor stem cells or their interaction with the microenvironment?
Yeah, I think it's a really important idea, and I think one of the challenges that we have is that we have rapidly evolved to a standard of care. So you have patients that for BLL they will get chemotherapy, and if chemotherapy doesn't work, they will shift to CAR T therapy, and they're sort of committed to a process that is going to be a very expensive process that has a good probability of not working for a long period of time, right? That maybe initial successes, but maybe failures. This, I think, has been a challenge for chemotherapy for a long time. Even though chemotherapy is a lot cheaper, but we give 4 or 5 drugs for B-cell malignancies, and we have been giving the same 4 or 5 drugs for 50 or 60 years, and there's no ability to really deviate very significantly for a lot of these therapies unless a patient can tolerate them. And it's very hard to know which of the drugs is having real efficacy in an individual tumor just because they're given as this sort of combination, right? That if you said to a doctor, okay, don't give this drug, you would say, well, that's crazy. That's what the therapies that we give. So I think I would say you have to have good models. You have to have models that allow you to say, okay, well, what happens, if we change the dose regimen? What if we actually, I would argue that maybe it would work better if you actually gave CAR T cells over several days, right? Not just, an initial big dose. Maybe you have an initial wave where the CAR T cells get killed, but then if you add another wave, you might actually, that is very difficult to do in a patient population, I think, as you mentioned. I think we have to find ways, we have to find models that allow us to change, to characterize MRD states, to look to see if we can see predictors of response in patients. Because if it's just, if we're just relying on the end clinical use, we're gonna be stuck in the same process. So I think we have to develop models, and I think those have to be combined with better ways to actually image and assess response in patients as well. Like being able to pull things out and look at immune profiling, looking at tumor cells, persistent tumor cells, even identifying. I mean, BLL, is more curative, but other leukemias like acute myeloid leukemia, like being able to understand what are the cells surviving, what is their state, how can we track it after therapy? These are important research questions.
Hello, Professor. Thank you for your very productive presentation. I have 2 questions. First of all, you talked about the combinational chemoimmunotherapy in your presentation. I want to know which of the cancers have, according to your research, which of the cancers have the best results and outcomes of survival for the simultaneous combinational chemoimmunotherapy? And as for my second question, you talked about the CAR T cells resistance with the Janus kinase pathway, JAK-STAT. Since in my PhD thesis, I am already working in the same thing about the silencing the STAT Janus kinase pathway and using the chemoimmunotherapy for TMBC cells. And we're getting good results too, but I'm so curious about can we use simultaneously silence the Janus kinase pathway and give the CAR T cells simultaneously so we have better outcomes with using them simultaneously? Silencing the Janus kinase pathway and simultaneously giving the CAR T cells so we block the resistance too.
Yeah, so in response to the first question, one of the I think, notable things that we found in our study is that it seems like the tumors that actually persist following therapy that may show intrinsic resistance to chemotherapy actually may be the best ones to combine with chemoimmunotherapy. Just to describe what I mean by that, what I think is happening is that you actually have chemotherapy that is hitting the target tumor cells. These cells undergo a senescence-like state and they start secreting factors. If you treated that tumor and it went away, then it's not secreting anything, right? You just have reduction to a small number of cells. So curiously, you actually need to have the persistence of a subset of tumor cells that allow the immune system to be acted upon and recognize it. And that may be an emerging theme with chemotherapy, that maybe the best thing to do is not try to eliminate the tumor completely, but actually try to make the tumor more recognizable so the immune system is better at coming in and completely eliminating it. So these tumors express BCL2, lots of it, they're very intrinsically resistant to chemotherapy, so they may be the ones that are more sensitive to chemoimmunotherapy. In response to the second question, yeah, this has been a real challenge because one of the issues we've had in silencing JAK-STAT and even interferon gamma is that the CAR T cells actually frequently need it to be active CAR T cells. So if you actually gave a systemic therapy, you may sensitize the tumors to CAR T therapy, but you also may block the ability of the CAR T cell to get activated to kill that target tumor cell. So we need ways to sort of selectively target the tumor cells initially and then come in with the CAR T cell. So you may actually be able to pre-treat patients with JAK-STAT inhibitors and then add CAR T therapy. That may be a better way to go.
Thanks a lot for your response.
Thanks for your presentation. And my question is, sorry, what's your thoughts about liposomal cancer vaccines?
Liposomal cancer vaccines?
Yeah.
I hope that they're still funded in the United States in the future. We will see. I think it's incredibly promising. But, we have half of our institute at MIT are sort of biological engineers that focus on liposomal formulations and have they helped in the generation of our own COVID vaccine, our Moderna vaccine, but also obviously the Pfizer-BioNTech vaccine uses similar modality. I think it's a very exciting modality. I think delivery is the challenge, but delivery is getting solved more and more every day.
First of all, thank you for your presentation. Unfortunately, I'm just a bachelor's student, so sometimes
That's okay. I was a bachelor student too.
It's just sometimes too hard for me to catch up the words, so sorry if I ask something that you have already mentioned. Yeah, I totally understood what you told about the resistance mechanism and how it happens in conventional anti-cancer drugs in some microenvironments, but how does it really work for monoclonal antibody drugs? Because they are smart and they are big molecules, generally in shape of protein, and they bind to, I guess, healthy immunogenic cells, then cause to attack to cancer cells or maybe multiple sclerosis or whatever it is.
Yeah, it's actually a very good question. So resistance to antibody therapy can absolutely be mediated by a mutation in the target. So the easiest way for a CAR T cell, a tumor to evade a CAR T cell is for the tumor to mutate the target of the CAR T cell, right? So if you don't have the target for the antibody, you write a very specific interaction, and blocking the interaction of the drug and its target, that's a primary mechanism of resistance. And if you did a screen in cell culture, that's what you would find, like entirely. And if you look at people, it's what you would find half the time or less, So I think that the relevant interaction there is not just the interaction between the drug and its target, it's what the drug has to do to actually kill the target. So in this case, the antibody can bind to the target, but it's not the antibody by itself that is killing the target. The antibody has to recruit a macrophage to come in and gobble that target tumor cell. So there you have a microenvironment that has to be operative. You have to have a macrophage be able to be present and active and get to a target site, and there are some sites where macrophages don't exist. And so for things like antibody therapy, if you think about MS or things like that, you actually have to have drugs that can get to the brain, right? And you have to have the right effector cells that are able to get there. And you actually have to have the right stimuli that are going to allow immune cell to be active to begin with. So you start seeing a whole other set of factors that become really important when you're treating a person, as opposed to treating a cell culture. And there are lots of different ways to model that, and I think we get at different answers in different ways. I think organoids are a great way to look at cell-cell interactions, for example, in different contexts. I think in vitro studies are incredibly powerful as well, so I'm not discounting that. I just think we need to think altogether about why drugs are failing in patients.